And so I was trying to make the point that when you are a microbe, you don't have those lovely internal organelles to rely on to do a bunch of your chemical and biochemical reactions. And so their cell membrane is particularly important because of that. And so this image is trying to get you to think about that yes, we tell you that membranes and cell membranes are really important for a permeability barrier. And certainly that's still true, but they have to do so many of their biochemical reactions in the membrane that they really do have a lot of proteins and enzymes and cofactors and such that are part of the cell membrane in order to run those many reactions. And then this last idea here is also really important because we're going to start getting into the physiology stuff in a bit here, and we're going to find that they need to develop essentially what's a proton gradient across their cell membrane. And when they can do that successfully, it sets them up well because that's a source of potential energy they can use to do something with that. And very often it's to make ATP, but they can do other things with it as well. And so like we use our mitochondria for that, a dedicated spot inside of our cells. They have to use their cell membrane for that. Okay, so cell membrane is really important. The other part of the microbe, of course, is the cell wall. I say cell wall, your book tends to say cell envelope. So this is a mock-up, very simple mock-up of big different, comparative similarities and differences between the gram-positive and gram-negative. You've already seen this idea in lab when you did the gram stain, where the gram-positive bacteria have much more peptidoglycan, many more layers of peptidoglycan than what you see with the gram-negative. So up to 90% of the cell wall can be made out of peptidoglycan. With the gram-negative, it's kind of funky. They have this outer membrane that's actually part of the cell envelope or cell wall. And then they have this space that's called the periplasm. So that's just referring to the physical space between that outer membrane and the cell membrane. And you can see in the periplasm area, there is a very thin strip of peptidoglycan. So only about 10% of a gram-negative cell wall is made up of peptidoglycan. And that's one of the huge differences that allows for the gram stain to work so well when you do that differential staining. Here's a little bit more detail. Again, just trying to show some of the major differences, and the peptidoglycan is the big idea here. And then the other big idea is that outer membrane showing up in the gram-negative cell wall. And you don't really have that at all in the gram-positive. Some very nice electron microscope pictures showing those. Here's another image. Again, you see the big purple stretch there in the top image. That's your peptidoglycan in the gram-positive. And then a very thin purple stretch in the gram-negative. And then you did do the acid-fast stain that has a lot of that glycolic acid lipid in the cell wall. We're not really going to get into the acid-fast cell type in this class. Again, I like this picture just because it's showing how busy the cells are. There's a lot going on there in terms of proteins and enzymes and all kinds of things going on in the cell membrane. This is a gram-negative bacteria where they're showing a lipopolysaccharide really stretching off of the cell wall. We'll go through that. And then you can see too in the periplasm that you do have, they're trying to show that there's lots of stuff in the periplasm area. So that's a nice reminder too. So let's think a little bit about the peptidoglycan. What is it made up of? And when you look at it and start to like bring it down to its simplest form, it's really two different sugars. One is N-acetylglucosamine and one is N-acetylmuramic acid. We abbreviate those down to NAG and NAM. And so they bind with each other, alternating nag-nam, 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 down these long, long chains of sugar derivatives. And so they alternate like that, nag-nam, nag-nam, nag-nam. Then we also have, as part of the peptidoglycan, some short stretches of amino acids. So we're going to see a picture that you have some amino acids kind of dangling off the NAM sugar. Excuse me. So there's L-alanine, D-glutamic acid, diaminopamilic acid in the gram negatives, but in gram positives, they seem to favor lysine over DAP. And so they'll have a lysine there. And then D-alanine. So those are the 4 amino acids that you see as part of the peptidoglycan, and they're bonded together coming off the NAM portion of the peptidoglycan. And so when you take that kind of all together, they say it forms a glycan tetrapeptide. Glycan is referring to the sugars, the nag-nam, nag-nam, nag-nam. Tetrapeptide means there's 4 amino acids, tetra for 4 amino acids. Okay, and then that just keeps repeating as it builds and surrounds the cell. Here's another mock-up of it. So if you look at this figure in the top left, what they've highlighted in yellow here is what they then blew up and put here. So you see the nag and the nam. And here's the nag and nam. And then you can see that there's 4 different balls here. Each one represents one of the amino acids. So you can see how 4 amino acids dangle off of this NAM, and then they bind to the 4 amino acids that come on the neighboring chain, also binding with the NAM. And so you see these chains of sugars, the nag-nam, 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 they're all laid down in these long chains next to each other, adjacent to each other. And then these amino acid tetrapeptides are linked to each other with the neighboring chains. And that's what keeps the stability of the peptidoglycan. They're also showing on this diagram some ways in which antibiotics mess with this. And so penicillin is famous for making it impossible for the bacteria to build new cell wall material. And so if you don't have an intact cell wall, you're in trouble. Questions on peptidoglycan? Here's another image of it. Just a slightly different look to it. Again, they're showing the nag and nam sugars. A little bit more detail. It's a beta-1,4 bond between them. And you can see on this diagram, they're pointing to the beta-1,4 and they're saying that that's sensitive to lysozyme. So lysozyme, I don't know if any of you've had any like human fizz or animal fizz, but lysozyme shows up in our saliva and in our tears. And so this is good for us because if we're eating or drinking contaminated food or water, we have lysozyme that can immediately start messing with the beta-1,4 bond of peptidoglycan. And so that's one innate response that we have to degrade bacteria as we're chewing. And of course, for your tears, it's important that your eyes obviously aren't getting attacked by pathogens. And then they're showing the 4 different amino acids. They call them peptide crosslinks. The 4 amino acids off of one NAM will bind to the 4 amino acids of the neighboring chain. So basically, when you look at it, kind of zoom out and look at it, it's really like a sheet of these chains of peptidoglycan with those peptide crosslinks between neighboring chains. And even though it's sugar, you think it wouldn't be that strong, but it is when you have that kind of formation like that. And very often, people will use the analogy as kind of like chainmail on a suit of armor. The chainmail idea that would protect from getting stabbed. It's kind of like that idea. And then sometimes, so we have those 4 peptides dangling off the NAM. And then they bind to the 4 peptides next door, so to speak. And then they found, especially gram-positive bacteria, they can have a few extra peptides thrown into the mix, right where it's binding. I'll show you a picture of that. So gram-positive sometimes put a little extra flavor on how they build their peptide cross links there. And then like gram-negatives tend to have basically 1 sheet of peptidoglycan and call it good. And then of course they build that fancy outer membrane. But gram-positives tend to have many, many sheets of peptidoglycan stacked on top of each other, all surrounding the cell. Here's another image. This one's comparing gram-negative to gram-positive in the sense of some of the little differences in which, what's in the peptide cross-link, and then here you have that inner bridge. Very often it's glycine, that amino acid. They'll just put a bunch of glycine together and then use that as part of their peptide cross bridge. And then this is kind of getting at the chainmail idea. You can think about chainmail in a suit of armor. It's kind of that idea. So if you take that and just keep building, blocks of this over and over and over, you end up with that really strong cell wall. And it takes a lot to break. When I was in grad school, we had to lyse open some bacteria, and we actually had to use a piece of equipment called a pressure bomb that would be forceful enough to burst open those cell walls. And the other way I've learned how to do it, but only with certain bacteria, when I used to teach BI 100 here, I wanted the students to be able to see DNA coming out of cells and precipitating so they could visually see it. But I didn't want to use all those nasty chemicals you typically have to use because they're non-majors biology, they're not biology majors. So I didn't want them working with really nasty stuff. So then if you take bacteria that are adapted to really high salt conditions, like, didn't you work with mannitol salt agar, right? Or no, you're going to do that. Sorry, you're going to do that. You're going to grow some bacteria on mannitol salt agar. And when you do that, it's high levels of salt in the agar, and only certain bacteria can exist on that and survive. And so I would take, there's another group of bacteria that can handle mannitol salt agar no problem. And in fact, they've adapted to like 20% sodium chloride in their surroundings. And so if you take those bacteria and pop them into pure water, they just explode because of the water rushing in due to osmosis. And that's enough of a force to cause the cells to burst open and release all the inner contents, including the DNA. So that was a really easy way for me to have them visualize DNA safely without using nasty chemicals. Another thing with gram-positives, because they have so many layers of peptidoglycan, they tend to have these lipoteichoic acids. Lipo just refers to the idea that they're anchored into the cell membrane, and then they go all the way through the peptidoglycan sheets. And then you can see that there's other proteins too that can be embedded in the peptidoglycan. So it does provide that kind of rigidity and structure, and making sure that the peptidoglycan is like attached and anchored to the cell membrane. Okay, now gram-negatives, they have to do an extra special thing. They put that outer membrane on the very outside, and they limit their peptidoglycan to basically 1 sheet. So they have this really fancy outer membrane. It's not the same as the cell membrane. It's not a phospholipid bilayer. It's not like that. We call it a lipopolysaccharide. Those of you who have taken Med Micro, that should sound familiar. So we'll dissect this. We'll look at some pictures and identify these different parts. So you have a lipopolysaccharide portion to the outer membrane. Then you have a periplasm. That's just the space, the physical space. And then you have usually a very thin peptidoglycan layer located in the periplasm. And then their cell wall, even though it has this so-called outer membrane, it's pretty permeable to small molecules. It's not highly selective, is how they usually put it. That outer membrane isn't terribly selective like you see with the cell membrane itself. So here's a mock-up of the gram-negative cell wall or cell envelope. So here you can see the few sheets of the peptidoglycan. And then here, the whole structure outside of that is called the lipopolysaccharide. And, well, I guess technically, yeah, it's the outer membrane that's called the lipopolysaccharide. So you can see that there's several components to the lipopolysaccharide. They've color coded it. So let me go to the slide that shows the color coding. So here's the color coding that corresponds to that. So the very outside of the outer membrane, the part that's dangling off furthest from the cell, is called the O polysaccharide, O-specific polysaccharide. So that's the outermost portion of the outer membrane. And then attached to that is the core polysaccharide in the pink. And then the lipid A is the yellow. And so when you look back, corresponds to that. So you have the O in the purple, the core, and then the lipid A. They represent the lipid A by these squares. Okay, and then you can see there's some fatty acids hanging off those. You can also see some porin proteins here. So these are channel proteins, and there's different porin proteins. They usually use different letters as designations, like there's OMP-A. OMP stands for outer membrane porin. So A, B, and so on. So they know the characteristics of lots of these porins. Then you also have some other proteins that can be embedded in that outer membrane. But that channel, the porins serve as channel proteins that allow for quite a few things to get through. And so let's, in words, what we were just looking at is the O. You don't need to know what they're composed of, but I've had students in the past say, what is an O-specific polysaccharide? If you really want to know, that's what it is. Core polysaccharide is composed of those components, but again, you don't need to know that. The lipid A, that's what's toxic to humans. So you might have, if you've taken Med Micro, you've probably heard about the LPS can cause problems to humans. If you have an intact gram-negative bacteria in you, it's not a big deal because the lipid A is part of an intact microbe. But if the lipid A portion of the lipopolysaccharide, if you get exposed to that in high doses, so let's say you take an antibiotic that kills a bunch of gram-negatives in you all at once, and those cells break open all at once, and you get exposed to lipid A, your immune system in some people, they go into shock, and it's not good. I've witnessed it happen to a family member, and luckily the doctors were there in time to catch it, and everything, went well. But that's something to be aware of. It's the lipid A portion that can be toxic to humans. It's technically called an endotoxin. We'll talk about, there's endotoxins and enterotoxins and exotoxins. We'll talk about those a little bit later in the semester and come back to lipid A. And then there's fatty acids that dangle off the lipid A. right? So back to that one. So that previous slide is in words, what you're looking at here in a visual drawing. And again, you don't have to know the intimate detail of all the parts of it. Know O, core, and lipid A. That's what I'm going for, okay? And then if you look back, whoops, not that one, this one. If you look back to this one, so what we just described is from here to here. So now we have to look at the second half of this outer membrane. And you can see if you just look along here, it's not as fancy. It looks more like a typical phospholipid bilayer. So that inner portion of the membrane, of that outer membrane, is not so fancy. That one has phospholipids, it has some lipoprotein that helps to anchor that outer membrane to the peptidoglycan so it stays stable and intact to the cell. And then those porine proteins I mentioned as well are part of coming through the whole outer membrane. So, okay. And then you can see the lipoprotein here is making sure that that portion of the outer membrane is linking into the peptidoglycan, so it's stable to the peptidoglycan. Then the other thing to think about is this periplasm. Before we get to the actual cell membrane itself, we have to think a little bit more about the periplasm, which is just a physical area. It's a very busy area. It is 12 to 15 nanometers wide, and it's very gel-like because there's so much stuffed in there. Lots of proteins and enzymes, lots of molecules coming in and out of the cell through that area. And so there's a lot going on in the periplasm. And don't forget the peptidoglycan is also there too. So that's why it's so gel-like, because there's a lot of activity in that region. It's kind of the last stop before the cell membrane. So it's an important area for the gram-negative. So here's a nice visual of a real gram-negative where you can see the outer membrane and then the very thin peptidoglycan in the periplasm, and then the cytoplasmic or cell membrane below that. Okay, so gram-positive, gram-negative, be comfortable with compare and contrast. You know, what are their similarities and what are their obvious differences? The makeup of the lipopolysaccharide is important as well. Questions on that material? No? All good? All right. Where did it go? Here. A little bit about archaea. So the domain archaea, I'm just going to say a little bit about them because their cell walls have a lot more variety than what we see in the domain bacteria. So the gram-positive and gram-negatives, you know, once you see one gram-positive bacterial cell wall, it's pretty much like that. Same with the gram-negatives. There's not a whole lot of variety. Archaea, though, it really depends where they were adapted to, where they were isolated from and adapted to, because their cell walls can have a lot of different components. So one type of cell wall is called a pseudo-peptidoglycan. Pseudo, what does pseudo mean? You say something pseudo, fake. It's fake. It's important you remember that. The pseudopeptidoglycan is not peptidoglycan. Pseudopeptidoglycan means fake peptidoglycan. It is not true peptidoglycan. If you look up the definition of peptidoglycan, this isn't it. All right, so the pseudopeptidoglycan does have a NAG component, N-acetylglucosamine, but it does not have NAM. Does not have N-acetylmuramic acid, and in place of it, it subs in NAT. Which in the notes, I think I have somewhere what that is, and you don't need to know it. But basically, it's not the same thing. NAG, it's the idea of NAG, NAT, NAG, NAT, NAG, NAT. You have that going on where it's long chains of these sugars alternating. But when you look up the definition of peptidoglycan, this is not it. Okay, it's NAG, peptidoglycan is NAG, NAM, NAG, NAM, NAG, NAM. Okay. And they have different linkages too. They don't do a beta 1,4 between the NAG and NAT. They do a beta 1,3, which is insensitive to lysozyme. Our tears and saliva don't touch it. But generally that's okay because although we have a few archaea that hang out with us and in us, they're not considered something to be concerned about in the sense of a negative impact on our physiology or health. So if you're curious, this is the idea of the pseudo peptidoglycan. And I guess you could say why they named it pseudo, because you see a lot of similar ideas here where you have the alternating sugars and then you do have these peptide crosslinks, but they have a very particular definition for peptidoglycan, and it has to include NAM to be officially peptidoglycan. So this is pseudo. So that's one type of cell wall that they find in the domain archaea. So that's what we just covered. Some archaea, instead of that, they just have cell walls of polysaccharides, just lots of sugars bonded together. Or glycoproteins, proteins, or mixtures of all three. And then that picture that you're looking at is called the paracrystalline surface layer, S layer for short. And a lot of archaea have this. In fact, the paracrystalline S layer is the most common so far of all the archaea they've studied. More archaea have this beautiful cell wall than any other cell wall type, even more than the pseudopeptidoglycan. All right, so this one wins so far. So obviously it is a pretty versatile, the paracrystalline S layer is a pretty versatile structure for them for a lot of different archaea to use in different habitats. Okay. So that's basically all I'm saying about archaea because they just have a lot. There's so much diversity there. That's all I'm going to say about the cell wall. Okay, I'm going to shift gears into the other slide set that's also posted in EDUCAT part 2 of the cell structure and function. So when you look at microbes, there's other things to consider besides the cell wall that can make up some microbes. And so one of the more famous things, I guess, to think about that can come off the cell of a microbe is their flagella. And so that is an amazing structure that some microbes only have one, some microbes have a whole bunch. And it's bizarre that they do that because when you look at the structure of flagella, it is not simple. So flagellum is singular, flagella. So I'm going to dissect it here in words, and then we're going to look at a picture of it. So you have the so-called motor protein, or motor portion, that's anchored into the cell membrane. And when you look at it, you'll see that it contains a ring, a so-called ring system. They look like rings. And then a central rod that goes through those rings. And then there's going to be special proteins called MOT proteins. I say MOT, maybe other people say MOT, but I have to say Mott because my one brother, his name is Tom, and that's Mott backwards. And my older brother used to tease my younger brother by calling him Mott all the time. So that's all I can think of when I see Mott is Mott. So I don't say moat. Anyways, there's these Mott proteins that kind of hung up next to the motor portion, and I'll show you that. And it's what's going to allow for the actual filament that sticks way out of the cell, that's going to rotate. So when we think about flagella in bacteria, they're different than what you've learned like flagella in sperm or with protozoa. Those whip around, they're very flexible and they whip. Bacteria took another approach. They're kind of like a propeller on a motorboat. They're helical in shape and fixed in that shape, and then they just rotate. So they don't whip. And it's a huge difference between what we see in eukaryotic cell flagella. And then there's these so-called flea proteins that they call them the motor switch. The flea proteins switch the direction of is the filament going to rotate in a clockwise or counterclockwise. So the flea proteins will shift the direction, will switch the direction. So all of that will then have a connection to what we call the hook. And then the hook connects to the filament, which is actually what you see when we look at tomorrow. We're doing the flagella slides. We're going to look at prepared slides of flagella, and we typically look under the microscope and go, "Oh yeah, look at the flagella." Really, what we should be saying is, "Oh yeah, look at the filament," because that's the part that sticks out of the cell. But we don't say that. We always say flagella. But you have to realize there's all these other subcomponents to it that you don't see when we do the flagella stain. So the filament is just made up of repeating protein subunits that are built and put together. And at least they name the protein something recognizable. They call those proteins flagellin proteins. Okay, so those subunits are called flagellin proteins. Okay, so that's all in words, kind of starting with what's anchored in the cell membrane all the way to the part that sticks out of the cell. Here it is in a visual. So here you can see the cell membrane. And then you can see these different rings. You don't need to know the names of the rings, okay? What kind of cell type did they draw here? Gram-positive or gram-negative? Negative. Yep, gram-negative. You have that periplasm with a very thin peptidoglycan and then this funky outer membrane. So you can see that there's rings that help support all of this through, in this case, the cell membrane all the way to the outer membrane. And then you have the rod going right through the center of those rings. And then you can see the Mott proteins, the big brown sausage-like structures. And then the flea proteins also there. And then you can see the hook region is just like a little adapter to make sure that all of this can then move the filament. Okay, so the filament, all those brown balls are just the flagellin proteins all put together. So you can see it's quite complex. Lots of flagellin protein is needed. And then that's going to build, that filament will end up being kind of a corkscrew look to it, spiral or corkscrew look. You know, it'll be rigid. And then what they're trying to show over here is the Mott protein or motor protein. They're trying to show that this plus minus idea here along the rings and the rods. And then the purple swoopy line, what they're trying to get across there is that if the microbe can build up, and again, here you see plus and minus across the membrane, anytime you see that notation, that's trying to denote that there's a proton gradient. Okay. And so they're trying to show that you have a lot of protons showing up in the peptidoglycan area, so you have a buildup of positive charge there, less protons inside. And so those protons want to diffuse through to even out their concentration across the membrane. As they do that, they go through the MOT protein. And once they do that, it creates the torque to actually rotate the filament. And so this will, the rods rotate, and that is transmitted through the hook all the way to the filament, and it causes a rotation. Some studies that they've done in the lab, they estimate 1,000 protons have to go through for one crank, which seems like a lot. So, I don't know. That's a lot. I mean, just look at this. This is a lot to have to build. And E. coli that you've worked with in class already, they have many, many flagella surrounding on, just stuck around their whole cell. They do this over and over and over again. Just seems like a lot of effort. And then it takes quite a proton gradient to get that to rotate. Questions on the flagella structure? Yes.
Is it only in gram negative?
No.
Is it like, is the structure
Uh,
significantly different from gram positive?
Do we have modal gram positives that are trying to come up with one? My brain's not working right now. Let me look into that and get back to you. Many, many, many gram negatives have flagella. Gram positives, I'll look into that. Gram positives also have other ways of moving, so do gram negative. So flagella isn't the only way they can move. It's the fanciest way and the most involved. This is, I think, this is from a previous edition of your textbook. I think they upgraded the picture to this, but it's basically the same thing. This doesn't have as much visual detail. I don't really like teaching off of this one, but if that helps you, you can certainly use it. And then here, they're trying to show how the bacteria, if they have many flagella, it's the most bizarre thing. They'll put all their flagella in a bundle and then use the whole unit, and rotate the whole unit. So they have to coordinate all their flagella like that, and then they'll rotate it, and then that can get them to, in this case, being propelled forward. And they actually, they don't have the word on here. So this is actually called a run, like running. This is called a run when they're going like this with their flagella. And then when they stop, their flagella fly apart from each other, and they call that the tumble. And so they say bacteria like this do a run and tumble motion under the microscope when you're looking at them, and they'll fly apart like that, and then they'll kind of let the liquid move them. And then when they feel they're in the right spot, they'll get all of their flagella coordinated again and start running, do the run again. And so it's amazing how complicated it is. And yet it's very successful for them. So I need you to know this word, both in lecture and lab, you'll need to know this word. Peritrichous is when you have many, many flagella pasted across, your whole rod shape. So E. coli is like that. And then there's some flagella that, like for this, they just have one flagella at one end. And you can see that counterclockwise will push them forward. If they rotate it the other way, it pulls them back. So it can be a push or a pull. And then this one, it looks like some bacteria can only do it in one direction, and then they wait for the liquid to kind of move them around a little bit, and then they'll wander off again. So there's slightly different strategies there. Here's some really nice pictures. So Peritrichus, again, like E. coli, pods of flagella all around their rod. Polar means at the very end of the rod. Lophotrichus, polar usually means just one. Lophotrichus means a tuft, like, 3 to 4 or so flagella, but they're all at one end. Peritrichus, it's all over. I mean, look at this crazy picture. So you go from polar, just one, to Peritrichus. I mean, that's just nuts. Look at how many flagella. That's a lot of effort. So when we do the flagella test with you in lab, we'll see how this works because if you were E. coli living in NMU's lab and you're given all the nutrients you want, you even get shaken, so you don't have to swim. We shake the tube for them so they're able to just kind of float around and get their nutrients as they shake by them. Would you want to build all this? Remember, microbes can purge really easily, and so I made the mistake of giving E. coli the sweet life over and over for years on end. I used the same E. coli line, so to speak. And then when I went to do the flagella motility test with the students, it refused to be moral because it just said, I don't need all this. I'm living the sweet life. And so they don't have to necessarily build all of that, which actually, when you think about it, it's a good strategy. Don't waste your resources on that if you don't need to move. Okay, but many microbes out in the real world, they need to move. It's a good advantage. Okay. Questions on the wacky world of the flagella? Okay. If you have your laptop, why don't you switch? We'll get into Educat and I can go over the exam. On Wednesday, we will get into other forms